Good afternoon to all. I'm Rafi J. Fornillos, a PhD in biology by research student, and I will be presenting a short seminar on Taeniasis and cysticercosis in the Philippines, history, research gaps, and way forward. I am pursuing research in parasitology, particularly in helminthic neglected tropical diseases such as schistosomiasis, but for my PhD dissertation, I'll be focusing on Taeniasis and cysticercosis. This short seminar will discuss briefly on the global burden of Taeniasis and cysticercosis, information on the causative agents of both diseases, which are taeniworms, and how they are transmitted between hosts and in the environment. Moreover, historical background and gaps on research on taenia, as well as the concept of One Health as the way forward, will be discussed. So to start, Taeniasis is an infection caused by the presence of adult worms in definitive hosts such as humans. There are three major species of Taenia that infect man, which I will enumerate in the next slide, that cause significant morbidity and mortalities in endemic areas. Moreover, cysticercosis is an invasive infection characterized by the presence of larvae in the musculature and nervous tissues of intermediate hosts such as pigs, cattle, and even humans. This kind of invasive complication is so far only associated to one species of Taenia. The major transmission route of Taenia worms from intermediate to definitive hosts is the ingestion of eggs and cysticerci larvae from eating contaminated meat. Sociocultural factors, such as the habit of eating raw or undercooked meat produced from backyard hog raising, is a major risk factor associated to Taenia infection. Considered as one of the 20 neglected tropical diseases identified by the World Health Organization, Taeniasis and cysticercosis are one of the few other more neglected tropical diseases where challenges in diagnosis, case finding, case management and treatment, as well as awareness of the disease remains unaddressed in many poverty-stricken communities. These two diseases have caused an estimated disability-adjusted life years, or DALY, of around 2.56 to 8.30 million, which are based on reports of epilepsy prevalence data records according to a 2015 WHO report. So this is usually associated to the severe and more life-threatening form of cysticercosis called neurocysticercosis. Taeniworms, just like any other parasites, are equipped with morphoanatomical features which are evolutionary derived to ensure continuation of their species, such as through production of resistant yet infective eggs, infectious larval stages, and reproductively active adult worm forms. Taeniworms are well known for their scolex with four suckers and a significantly long worm body. This worm's entire body length is characterized by repeating metameres of proglottids, which may appear in various reproductive maturity stages and contains all organs, particularly the testes and ovaries. Taenia worms extract nutrition from their host by penetrating its scolex in the intestinal mucosa of its host, and eggs from gravid proglottids, which are carried along with the feces and expelled to the environment through defecation. The three species of tapeworm that primarily infect man, namely Taenia solium or pork tapeworm, Taenia saginata or the beef tapeworm, and Taenia asiatica or the Asian tapeworm. The common diagnostic morphological feature used to identify adult worms are the presence or absence of hooks on the rostellum of the worm's scolex. Taenia solium have distinct hooks, while rudimentary hooks are observed in Taenia asiatica and no hooks are seen in Taenia saginata. Transmission of Taeniasis occurs when a human ingests the cysticercus present in the pork or beef or even liver of an infected intermediate host. Cysticercus exists and the invaginated protoscolex emerge and attach on the intestinal mucosa for feeding. Intermediate hosts get infected by ingesting food material contaminated by human feces where taenia eggs or gravid proglottids are present. On the other hand, cysticercosis develops in a human when eggs of Taenia solium are ingested. Eggs further form as cysticerci larvae and invades muscles and nervous tissues. So for the epidemiology of Taeniasis and cysticercosis, Risk factors such as cultural practices involving eating raw meat with poor food safety measures, complexity of food network and logistics, poor sanitation and hygiene, use of contaminated water in hog raising, lack of awareness to the disease, and the unavailability of medical care due to geographic isolation and other socio-political hindrances provides a conducive environment for a continuous transmission of these two diseases. These risk factors should be addressed to cut the life cycle with the goal to lessen or even eliminate morbidity and mortality associated from Taeniasis and cysticercosis. So, what has been done in the Philippines? Taenia research in the Philippines is scarce and definitely merits attention. There is a limited number of publications available reporting sporadic efforts to determine prevalence in selected endemic provinces in the country, and the early stages of parasite identification by experimental infections and detection of eggs in commodities such as fresh produce are few of the topics depicted in the literature. For example, so far, based on intensive literature search, there are seven researches only that are available from 1978 to 2017 that focused on prevalence surveys through stool examinations, serum ELISA, and stool PCR, and reports of prevalence vary since these techniques have different specificity and sensitivity rates. Populations targeted were primarily school-aged children, or SAC. Early stages of humanitarian research in the Philippines focused on clinical trials of drugs as potential anticystodal agents, as well as on serendipitous discovery of endemic areas through stool surveys. For surveys involving animal and environmental samples based on intensive search of available literature, there are three researches only that shed light on Taenia identification through experimental infection and in detecting Taenia eggs in soil samples and in fresh produce, which definitely provides significant information on the parasite's endemicity and transmission in certain parts of the country. As some say, with scarcity comes opportunity. Research is a powerful tool in order for public health experts to have an informed decision. A systematic prevalence survey, genetic studies on Taenia hybridization, improved diagnostics, disease modeling incorporating geospatial and temporal data are considered priorities for our country to have an efficient and effective surveillance system for both Taeniasis and cysticercosis. As we face the new roadmap imposed by the World Health Organization to target control and elimination of most neglected tropical diseases, including Taeniasis and cysticercosis, by 2030, One Health approach, which incorporates human, animal, and environmental health, is needed to ensure the transmission of such diseases are completely interrupted. We need to adopt this multifaceted approach of One Health, since by nature, most neglected tropical diseases covers the biological, environmental, sociopolitical, and cultural dimensions of the society, and lack of attention of these aspects favor transmission, further strengthening the association of poverty and infectious diseases, a cycle that almost 1 billion of people in the world find it hard to escape and break the chain. We need to incorporate advances in diagnostics, the use of GIS or geographic information system to determine places where transmission is possible, and to further educate the population that these diseases are highly preventable and curable if diagnosis is made at an early stage. We hope to control, eliminate, and eradicate these diseases to ensure a healthier mankind by forging strong and sustainable intersectoral partnerships, the very Waterloo of NTDs. These are the references I used in this presentation, and I would like to thank you all for your attention.